Hello everyone, this is lecture 3 from BE544. In today's lecture, we will deal with QuPath. As mentioned in the previous lecture, QuPath is software for bioimaging analysis. It can handle whole slide images, annotate it, perform different diagnosing on it, such as classification and detection. So you simply will navigate to this link and download the QuPath for your operating system. For example, if you're using Windows, you will find download for Windows. It will start downloading and then you need to install it. After the installation, you will find a shortcut for the application on your desktop, or you can search for QuPath in the search bar or Windows menu. The icon for QuPath will look like that. When you open it, initially you will find this UI. It will ask you to choose the theme and check for the updates. You can keep it like that or select the configurations you need and then you get started. If you need this window to show up again, you keep the tick here, show this on startup, and if you don't, you can cancel it. This is the interface of the application that we are going to deal with. So initially we need to create a project so we can import different whole slide images in that project, perform different operations, and so on. So you'll press on create project, create the project. It will require to select a folder. So we have selected the folders and you press on select folder. It will appear here. You need then to add different images to that project using the button add images. And if you close the QuPath and open it again, you can use open project if you have created it earlier, and you select the folder that contains the different items for that project. For today, we will use BACH dataset. It's a dataset for breast cancer histology images, which is public for research purposes. You will find the download links for the different items for that dataset. We will need the first one to download. So you will press on download here and it will start downloading. Then it's compressed. Then you need to uncompress or extract the different files from it. After downloading the file and extracting it, you will find two folders. One is named photos and the other is named WSI, that refers to whole slide images. If you open the photos, they are extracted in 4 classes, but currently we will need to use the whole slide images. They provided the whole slide images and also annotations, which are abbreviated in XML, but currently we will use only the whole slide images for this lecture, where the extension is .svs. We will press on add images and you can drag drop the images here, the SVS files, or you can simply choose files. Let's sort by name and let's select multiple SVS files, for example 5 files. They will appear here, and then you select the image type to be bright field H&E, because if you went to the source of the dataset, you will find that it's hematoxylin and you assign dataset or H&E, and you can keep it like that or say no rotation and press import. It will start processing the datasets or the SVS files, and you can find them under the structure of the project, or it's a created project, and different thumbnails will be displayed here for each of them. When you double click on any of the SVS files, it will open. You can decrease the size of that sidebar. You can zoom in and zoom out, because as we said, we are dealing with pyramidal structure. So the more you magnify or the more you zoom in, the more details you are getting. So let's zoom in. For example, the pink purple parts here or regions can refer to nuclei. If you open another slide by double clicking on it, you can zoom in to get the fine details. As you see here, some nuclei, white regions can refer to adipose or fat, and cytoplasm, and so on. The next step, we need to change the brightness and the contrast of the displayed image by clicking on that icon or pressing Shift C. You will find here a menu for brightness and contrast. As you see here, they are displaying by default the original image, but you can only, as we are dealing with hematoxylin and you assign SVS or whole slide image, you can display parts of the image. So we can display only the hematoxylin by clicking on the show icon next to the hematoxylin, you will find that only nuclei are displayed, and when you press on U sign, you will find the cytoplasm and the extracellular structure will be displayed. You can also press on show grayscale to display a grayscale image. So by looking, you will find different nuclei here. You can also change the channels minimum and the maximum. By decreasing the minimum, you are getting more contrast, and increasing the maximum, and so on. So by this manipulation, you are getting the nuclei visually here. Or you can press on auto, it will calculate it automatically, as you see here. You can also press on invert background, so it will be in grayscale, but it will change the black to white and vice versa. As you see here, the nuclei are converted to white and the background is converted in black. Let's return it to the original and uncheck for both of them. You can close that menu. So visually you can play with the brightness and contrast to visualize the different nuclei for an image, or the cytoplasm, or adipose, or the extracellular structure as you need. The next step, we need to perform manual annotation, and the usage of manual annotation here, as mentioned in the previous lecture, we can use these manual annotated from us or from a clinician or a doctor to provide it to a model that can later learn how to automatic segment or automatic annotate the regions. One of the reasons can be because the image or the whole slide image is very large. It can be more than 20K by 20K or more. It can reach 200 or 300 and more. As you see here in this image, as you go further, you are getting large numbers, 23K by 16. So it will be tedious for us to annotate the whole slide image and assume you are getting a big data, for example, 10K patients or 10K whole slide images. It will take longer to perform manual annotation for the whole slide images. So we can annotate different parts from different patients and then provide these annotations to a model that can automatically annotate all of these whole slide images. Also, we can separate between tumor and non-tumor parts if you are dealing with that, or separate nuclei from the image. Then we can use these annotations to provide it later to a classifier that can learn from the patterns from the tumor and the non-tumor, and then when we apply another tile or patch to that classifier, it will predict that it's a tumor or non-tumor. It can predict the severity of the tumor based on the annotation we are dealing with. So this step is crucial and requires concentration in the annotation process. The reason behind that is that assume that region is a tumor and we are annotating it to be a tumor, and then the annotator took a large region that consists of tumor and non-tumor parts, and later when we provided that to the system, the classifier or the model is misled because he got data that it thought to be a tumor but contained non-tumor parts. So we need to focus on the annotation, or the doctor needs to focus on the annotation process to be precise and concise. So in this bar, there are different annotation tools such as rectangle, oval or ellipse, line, polygon, polyline, brush, and wand. So let's start working with each of them. For the rectangle, you can simply press on it and you can select the region of interest you are dealing with or you need, for example, that part. Once you have drawn that, it will automatically return to the move tool, and if you need to draw another one, you can press on the rectangle or use the shortcut R to select another rectangle. Once you created another rectangle, the old one will be deselected and moved to red. And after a while, I will show you what are the different colors and the classes to put for each annotation. To select that again, you can simply double click on it. It will deselect other annotations. And if you don't need to select any of the annotations and go outside that region, you double click in the outside region, it will deselect both of them. You can also select it and delete using the delete button. You can also delete that by pressing the delete button. You can also use the oval or ellipse. You can draw a line. You can draw a polygon, and polygon here can be performed in two ways. You can put points and it will connect them using lines. For example, you press a point here, another point here, another here, and so on. As you see here, it draws a polygon. Once you are clicking, it will connect lines. Or you can simply continue clicking and drag the mouse. For example, like that, it will draw different points. And when you are done, you can release the click or the left click. To close the polygon, you can double click on the last point, and this is our polygon. You can deselect it by clicking double click outside the area. Also, if you need to move any annotations to any position, you can simply select it and left click on the annotation and drag to the region of interest you are needing. You can also resize it from the square points that bounds the annotation, as you see here. You can also do the same for the polygon by selecting it first, and then you can simply change the position of any square point like that or like that. You can use a polyline. For example, if we need to make an annotation here, it's similar to the polygon, but it doesn't close it. So you can simply double click here and it will keep it an open polygon or polyline. So another example, you can start to annotate that region, and when you get closer to that point, which is a starting point, it's look like a magnet, so you can make a closed polygon like that. Also, you can drag any of these square points or drag the figure or the annotation like that. You can deselect it. Also, there are two advanced tools, which is the brush and wand. So let's assume we need to select this white region or this adipose region, for example. So we can select the brush tool and start to select the region by continuing clicking and moving around the area. Assume you have finished and you need to increase that region to include also that white part, but after you zoom in, you can zoom in simply and be ensured that you are selecting the ROI or the annotation and you are selecting the brush tool. From inside, click and continue. It will continue to include that region like what you are doing now. Assume you need to refine or find the detail here, you can zoom in, and also from inside you can start to include that region. But if you didn't select it and selected the brush tool from inside to outside, it can also include the region you have annotated. But don't do that from outside to inside like that. It will draw another annotation. What about the wand tool? Let's first delete that annotation and use the wand tool. It's more sensitive more than the brush, especially if you have difficult shapes. So let's start annotation. Ensure that you are selecting the wand tool and you can simply press. It will automatically outline the region. Once you are getting closer, it will increase the region. It's based on the brightness, intensity, and the contrast, so it's more sensitive to the different regions and intelligent than the brush tool. You can consider it as a fast tool, so in less time you have annotated that region. You can also annotate here, so once you are getting closer, it will start to annotate the adipose or fat cells. Also, if you pressed on the wand tool here and started from inside to outside, it will include the annotation or the new annotation to the old one. As you are seeing here, it will select it automatically and include it, as you see here. You can get closer and press from inside and continue like that to select the other parts of the white region, as you see here. So assume we need to annotate that red part. It can be an artifact in the stain, so we are using the wand tool to include it. But assume during using the wand tool you have made a mistake like that and you need to remove that adipose. You don't have to remove the whole annotation, especially if you have annotated a long portion, you can simply press on the Alt key, and inside that region you continue annotation. The Alt key here will start to deselect the region you are highlighting. So it will deselect and minimize that selection as you see here. And after you finish, if you need to include another part, you release the Alt key and start annotation from inside to outside. Let's move to another example. Assume we need to select the nuclei here. Let's select the wand tool to start selecting that boundary region of nuclei. And we need also to include the inner pink part here. So let's select it. And we need to deselect the adipose region here, or the white region here. So we will press the Alt key, and from inside we will deselect the adipose region and select only the region inside here. You can zoom in to include the parts and exclude the outside here. You can refine the details. So assume that annotation, it includes the inner part and outer part. So assume we need to select the white region here to be in another annotation. So we have the first one in the red part and another one for the adipose. So let's select the brush tool. If you pressed on the adipose, it will start to annotate and include the adipose, but it takes another part from the nuclei, and we don't need to include that. So simply you can annotate that region, and before releasing the left click, you press on Ctrl Shift. It will automatically select the unselected regions. So it will calculate the boundaries that are not selected and select it for you. So this is another annotation. So there is no now overlapping between the first or the outer one and inner one. Let's take another example. Assume we need to annotate that region or the nuclei and the inner part separately. So let's select the wand tool for that and then deselect the inner part. This is the first annotation. If you click the outside, this is the annotated part, and we need to select this region only. So let's select, for example, the brush, and from the non-annotated part we start to annotate it. And before we releasing the left click, we press on Ctrl Shift. It will only annotate the non-annotated part. If you click the outside, these are two different annotations. Let's switch to the annotation panel. All of the annotation will appear here. As you see, if you double click on any of these annotation, it will directly move to that annotation. For example, if you double clicked on the circle, it will display it. If you double clicked on the last annotated part, it will move directly to it, similar to that. So let's select all of the annotations and delete it using the delete button. Click on yes. So we have only now two annotations. As you see, both of them are in red, but you can set a different class for each of them. For example, assume that part is nuclei. So we will select that part and press on nuclei and set class. It will directly be colored similar to the color of the nuclei, which is green. And for the inner part, let's select other, similar to that. If you click the outside, each of them is colored in its class. So for the green, they are referring to the nuclei, and for the orange-like color, it's for the other. You can fill and unfill the annotation with these colors using that button that looks like that. You can change the intensity of the colors using that scroll bar. Also, you can show and hide the annotations using that button like that. So you can put a boundary and fill the regions. If you enter to the hierarchy part, you will find each of them is separated. So if you need to combine a set of annotations together, you simply go to the annotations, draw a square or a circle or any annotation part that includes the annotations you need, like that. You can set a class for that annotation to be region. But if you went to the hierarchy, you will find the three of them are also separated. To combine that together, you right click on the larger region and select insert into or in hierarchy. If you went to the hierarchy, you will find that the annotation, which is the outer one, includes the first one and the second one. This is considered organization part for your annotation. If you went to the image panel, you will find the hematoxylin, eucine, residual stains, 3 stains. Each of them has a vector, 3 values, which is calculated automatically from the application or the QuPath. These vectors can be used to distinguish between the eucine and hematoxylin as we saw earlier here. You can double click on each of them and modify the numbers manually, or you can calculate it also automatically on a specific region from Analyze, Estimate Stain Vectors. It will display an interface like that. You can press on Auto to calculate the new vectors and press on OK. It will require a new name. You can set it as H&E estimated. It will update the numbers. If you went to the hematoxylin and eucine and displayed only the hematoxylin, it will display something like that, and for the eucine it will display like that. Let's return it to the original. Let's select a larger portion, and based on it we will analyze estimate stain vectors. If you found that warning, you can press on yes and then press on auto. It will declare and set the new estimation. Click on OK and press on the new name. It will update the vectors, and you can also select the hematoxylin and eucine or display the original one. By playing with that, you can handle the different vectors that can split between the eucine and hematoxylin, and the more you get a region, the more sensitive and accurate for the vector. Assume we need to select only the nuclei or the cells in the whole slide image. So instead of annotating each of these cells, QuPath provides a cell detection tool. From Analyze, Cell Detection, and press on Cell Detection. Let's keep these parts and configurations as it is and press on Run. It will give an error because there is no region that we need to select the cells into it. So let's make a rectangle like that and press on Analyze, Cell Detection, and Cell Detection, and then press on Run. It will start to run, and the more region you are selecting, the more resources from your PC or laptop will use. As you see here, it has selected all the cells based on the intensities of the vectors we have calculated or calibrated, and if we zoomed on each of them, you will find each nuclei is surrounded by an expansion or cell expansion, and this parameter is defined using 5 micrometers. So we can decrease that value, for example, set it to 0.1 and press run again. It will select the nuclei with a simple expansion of 0.1 micrometers. Let's select a small portion and run cell detection again with 0 micrometers. It will display only the nuclei. If you zoom in, you can find some artifacts in the image, like that one. It can be non-nuclei and refer to another thing or an artifact in the stain. So you can simply increase or decrease the threshold, for example, by changing it to 0.2. As you see, this portion is deselected, so the threshold here, as mentioned, the nuclei must have a mean intensity more than or equal that threshold. You can also define the minimum and the maximum areas for the nuclei. So for the minimum area, it's 10 micrometer square. So if you increased that to 50 and run the script again, it have deselected all of the nuclei that is less than 50 micrometer square. Let's set it to 25. It will look like that. If you set the cell expansion to 2, for example, it will include, as mentioned, the nuclei and the cell or the expanded cell. But if you didn't select this option, include cell nucleus, and run, it will display something like that. It will not separate both of them. It will include the cell as a whole with an expansion of 2 micrometers. You can play with the different options, and if you forgot the meaning of any of these options, just hover on it. It will display a legend for the meaning and the target of each configuration. So let's set it again to 0 and include the cell nucleus and run again. So as you see here, in this region, there are more than 400 nuclei, and if you went to the hierarchy, you will find that the parent, which is the rectangle we draw, and inside it there are different nuclei here. If you clicked on any of them, it will go to that nucleus. And for example, if you went to that one and we didn't like it, or it's an artifact or something, we can simply delete it like that. So it decreased by 1 from 427 to 426 and so on. You can export the results you got for the different nuclei from measure, and then you press on show detection measurements. You will find each nucleus here with the different parameters. For example, the area, eccentricity, standard deviation for the OD, and so on. You can save them. Let's write sample.txt and then save it. If you open that file, it will display the different parameters for the different nuclei. You can also open it from Excel. Simply drag drop that file into Excel, and then you can perform the different operations if you need on the detected nuclei. Let's move to another tool, but first let's delete the object. If you selected the object and pressed on delete, it will ask you to keep the child objects. If you need to keep them, press on yes. If you need to remove all of the annotations including the detected nuclei, press on no. It will remove them. Let's draw another rectangle. Go to analyze, cell detection and positive cell detection. As you see here, it's similar to cell detection, but they included the intensity threshold parameters to split between the positive intensity thresholds from low, moderate or medium and high. It will spread between two classes. One is positive, one is negative. So if you kept the default parameters and press on run, you will find here that it selected different nuclei, more than 2000. If you went to the hierarchy, some of them are positive and others are negative. If you removed cell expansion and press run again to select only the nuclei, it will select this region. The red is positive while the other one is negative. So you can get the benefit of the positive cell detection by playing with these configurations to separate between tumor cells and non-tumor cells, cells with high intensity, cells with low intensity, and compare between them using a model or something. If you want also to show detection measurements, you will see the classification, where the first is negative, the second is positive, and so on. So cell detection and positive cell detection are similar, but for the positive cell detection, it's make binary classification based on the intensity. Let's delete that part. It will ask again to keep. Let's press on no. The last tool we are going to deal with is the pixel classifier. Go to classify, pixel classifier, and train pixel classifier. If you pressed on live prediction, you will see nothing. Why? Because the pixel classifier requires some annotated parts that can learn from them. So let's close it. Let's, for example, separate between the nuclei or the cells with nuclei or the portion with nuclei and the cytoplasm regions. So let's select the one tool and annotate that part. This also. We can also select from inside to outside this region. If you went to the annotation, you will need to set this annotation to a specific class. So for example, let's select both of them and then select a nuclei and press set selection. You can press on auto for the future, so they can be automatically classified as nuclei. So if you selected that part, it's a nuclei, and that part also is nuclei, that region, that region, that region, and this region, this also. And let's select another class, for example, other. Let's select that region, this region. You can simply also use the brush tool. Let's select the one tool and select that region, this region. The last thing, you need to avoid the outer region, assuming this region should be ignored in the classification. So you press on ignore and annotate that region. You can also annotate that region, so it should be ignored. You can also annotate part of this region to be ignored here also. So you have different regions to be ignored. So let's start the classification and check train pixel classifier and press on live prediction. When you press on live prediction, it got most of the region is ignore and a little is nuclei and other. For sure this is wrong because the ignore part of the whole slide image is very small compared to the other parts. So you can simply go to features and press on edit, and for the channels select hematoxylin and use sign because we need these two features to separate between the cytoplasm and nuclei. Press on OK. If this issue appeared again, you will need to select a larger portion. So let's select that region to be the nuclei and change the class to be nuclei and ensure that we are selecting auto. Select that region also to be nuclei, that region also here and here and here, here and here and here. You can also for the other to select that region, and let's make the prediction. It have predicted that some nuclei and some other. You can also edit the features to be the iosine and hematoxylin. It have updated the different regions. So if you zoomed in, you can find it started to classify each pixel to be an ignore or nuclei or other. And if you have other classes, it can also classify them. The more you are annotating and the more precise regions you are annotating, the more that classifier will learn and produce accurate results. Maybe it learned from that region and misleaded, as we mentioned earlier, because we have included some nuclei regions here. But as an overall, you need to increase the number of annotations or annotated regions, and this region should be concise and precise. The pixel classifier is not limited only to separate between cytoplasm and nuclei. It can separate between tumor and non-tumor. So you can, if you are a pathologist or you know how to separate or identify visually between them, you can simply annotate regions with tumor and regions with non-tumor. So you can say simply there is no non-tumor here. So to add a class, you can right click, add a class. You can say non-tumor and the non-tumor appears here. You can double click on it and define the color, for example that color, and press on OK. So if you selected that and selected any region here and clicked outside, this region, if belongs to the non-tumor, should be annotated like that. And for more projects, you can navigate to the tutorials of the QuPath and navigate to any of these projects or tutorials and start working with them. For example, pixel classification. You can follow the guidelines here and start working and understanding the different concepts and steps using QuPath. So in this lecture, we have dealt with QuPath annotations such as rectangle, brush, and wand tools, how to create, open, and add images, how to separate between the hematoxylin and iosine stains and other stains, how to auto-tune the vectors of the hematoxylin and iosine using the stain vectors calculations. Then we dealt with cell detection, positive cell detection, and pixel classification. Finally, we knew how to deal with more projects and tutorials from QuPath website.